Hello everyone, my name is Derk. I'm a PhD student in the lab of Judith Klumpman and Ginny Farias. And today I will discuss one of the projects that we have been working on in the lab, which is titled Hobbs Disruption Impairs APP Trafficking and Processing, thereby promoting the exosomal secretion of APP-CTFs. And this work has been published in the beginning of 2026 in Neurobiology of Disease. In our lab, we are interested in the organization of the endo-lysosomal system. So the endo-lysosomal system is essential for multiple processes, such as protein transport, signaling, but also the degradation of macromolecules. Lysosomes are known as the main catabolic center of the cell, while endosomes are mainly involved in recycling of proteins. So why are we specifically interested in the endo-lysosomal system in the context of neurological disorders? Well, during neurological disorders, there are hallmarks such as swellings of endosomes and lysosomes. We find aberrant levels of endolysomal proteins. And lastly, what we often see is that there's impaired degradative capacity of lysosomes in these diseases. And one of the diseases that has all of these different hallmarks is Alzheimer's disease, which is still the leading cause of dementia worldwide. And one of the key players in this disease is amyloid precursor protein. Amyloid precursor protein is a transmembrane protein and is mainly known because of its role in the pathogenesis of Alzheimer's disease, since APP can be processed via base 1 in the amyloidogenic pathway, leading eventually to the formation of neurotoxic C-terminal fragment C99 and amyloid beta, which is present in amyloid beta plaques, which are known to cause neuronal death and also synapse loss. On the other hand, APP also has beneficial functions for the cell. So APP can be processed by NM10, leading to the formation of soluble APP alpha, which is actually known to be neurotrophic and promotes neuronal health. So in order to understand APP processing, it's also important to really know about the transport routes of APP. So APP can be either transported directly from the Golgi towards the plasma membrane or to early endosomes. In early endosomes, APP can recycle back to the Golgi, or it can be targeted for degradation into late endosomes and lysosomes. And what we see at steady state is that APP is mainly present in the Golgi apparatus, early endosomes, and the plasma membrane. At the plasma membrane, APP can be cleaved by non-amyloidogenic secretases such as ADAM10. So actually, the presence of APP at the plasma membrane is thought to be neuroprotective. However, if APP gets stuck in intracellular sites, amyloidogenic processing can occur. And therefore, it's really important to promote certain mechanisms that stimulate the degradation of APP or to stimulate its recycling back to the TGN for it to subsequently be transported back to the plasma membrane. In our lab, we are very interested in the Hobbs complex. So the Hobbs complex is a 6-subunit tethering complex, which is involved in the fusion of endosomes with lysosomes. However, we know little about how it is involved in the trafficking of key neuronal proteins. And why is it so important to study the Hobbs complex in the context of neuronal health? We know from previous studies that mutations in Hobbs subunits can actually cause neurodevelopmental disorders. And what we see in these neurodevelopmental disorders is that there are accumulations of endosomes and lysosomes, which is very reminiscent of what we see in Alzheimer patients. Moreover, recently, Hobbs dysfunction has also been linked to Alzheimer's disease itself. So therefore, we wanted to address what the role of the Hobbs complex is in APP trafficking and processing. And for this, we first focused on the question how Hobbs dysfunction impairs APP transport. So for this, we made use of already established Hobbs knockout cell lines. So in these cells, we depleted one single subunit of the Hobbs complex, thereby impairing its function. And in these cells, we overexpress APP with an N-terminal tag. And what you can see in wild-type cells is that you have tubular compartments and also fascicular compartments, like round in shape. And they are generally not too big in size. However, if we deplete these cells of single Hop subunits, we see that the compartment size largely increases, and this is also exemplified on the right. Then we wanted to check what are these compartments then. So we check with a panel of endo-lysosomal markers to see what it correlates best with. And one of these markers is the late endosomal marker LAMP1. And what we see if we deplete cells of the Hobbs complex, we see that there's an increased colocalization with this late endosomal marker. We also further checked with immunoelectron microscopy, in which the black dots indicate the halo tag of APP. And we see that APP is really drastically present in these late endosomal compartments with a lot of vesicles inside the lumen of these compartments. And therefore they are also called multivesicular bodies. So really to dissect where and when we see defects in APP trafficking, we use the RUSH tool to check APP trafficking in HeLa wild type and Hobbs knockout cells. And in brief, how this tool works, so we have APP, which is coupled to a fluorescent tag and streptavidin binding protein. We co-express this with an ER-specific hook protein, namely streptavidin coupled to the ER-resident sequence KDEL. And this leads to retention of APP within the ER, and this is also what you see in the photo on the right. However, upon addition of biotin, the interaction between APP and this hook protein is released, and therefore we can follow its transport pathway into the biosynthetic pathway and subsequently also its recycling and transport in the endo-lysosomal system. And this is nicely exemplified over here. So you see this accumulation in the Golgi apparatus, and after that APP gets nicely secreted into post-Golgi compartments. So then we checked how this relates to APP trafficking by checking with the Golgi marker GM130, because we see APP being stuck in endosomes. We wondered, is APP recycling back to the Golgi impaired in these cells? So in wild type cells, you can see after 1 hour after release, there's nice overlap with this Golgi marker, but also 8 hours after release, you still see that there's quite some overlap with the Golgi marker GM130. So that indicates that there's continuous recycling of APP from endosomes back to the Golgi. Not only that the release is normal, but also that this recycling pathway is working normally. In Hobbs knockout cells, however, we see that APP still is able to exit the Golgi normally 1 hour after release, so you see a lot of colocalization with this Golgi marker. However, if we look 8 hours after release from the ER, you see that APP gets stuck in these late endosomal structures, and that the Golgi apparatus is completely devoid of any APP signal, which is an indication that APP is not able to recycle back to the Golgi apparatus. So then we wanted to ask the question, like, does Hobbs dysfunction then also influence the colocalization with amyloidogenic secretases? And for this we focused on base 1 first and checked its colocalization in HeLa cells upon overexpression. So what we see in wild type cells is that APP and base 1 are nicely segregated into distinct carriers. However, if you then deplete these cells of Hobbs subunits, you can see that the colocalization of APP and base 1 is really drastically increased, indicating that base 1 also accumulates within the same late endosomal structures as APP. So then, does APP processing occurs in these late endosomal structures? For this, we used an immunoblotting method to check APP processing. And we used an antibody directed against the C-terminus of APP. And this antibody can recognize both the full-length APP as C-terminal fragments derived from both non-amyloidogenic and amyloidogenic processing. And what we found is that in wild-type cells, you see quite some signal for the full-length APP, but basically there's little to no APP-CTFs to be detected, which are the C99 and C83. However, if we look at Hobbs knockout cells, you can see that both fragments like C99, so the amyloidogenic APP-CTF, and C83 can be both detected. So indicating that there's indeed increased processing or increased accumulation of both fragments in these late endosomes. But then what about primary neurons? So we wanted to validate whether the same thing was happening in these cells. So to impair Hobbs function, we applied a different approach instead of using knockout models. So for this, we overexpressed OR3A, which is a SARS-CoV-2 accessory protein, which is able to bind to VPS39, one of the subunits of the Hobbs complex. And thereby it has been shown to really impair endo-lysosomal fusion that is normally mediated by the Hobbs complex. And what we find in wild-type cells is that APP is generally slightly polarized to the axonal domain, meaning that there's generally more APP in the axon versus the dendritic domain. However, if we look into Hobbs-depleted cells, Hobbs-impaired cells, we see that there's an accumulation of APP within puncta, so similar to what we see in HeLa cells. And that surprisingly, this accumulation really only occurred in the somatodendritic domain of neurons. So indicating that this local processing of APP by base 1 mainly occurs within this domain. So then we wanted to check indeed whether there's also more processing within this domain. So we focused on APP processing within the soma and in dendrites. And for this, we applied an alternative approach in which we tagged APP both at the C and N-terminus site at the same time. So if you would have no processing, you would see high co-localization between the N and C terminal tag. However, if you would have processing, the soluble fragment of APP can be secreted, and then subsequently you have less co-localization between the N and C terminal fragment. And also in our case, what we would expect is that we have a lot of C-terminal fragments and therefore a lot of compartments with only the C-terminal tag. And this is exactly what you see in this image in the middle. So on the top, you see that there is a lot of co-localization between the N and C-terminal tag. However, if we look in the cells in which we impair Hobbs function, we see that there are a lot of puncta which are only blue, which correlates with the M neon green or the C-terminal tag of APP. So this is an indication that we have a huge accumulation of APP-CTFs in Hobbs impaired cells. So we have processing into these CTFs and the cells is not able to degrade them. Then we were wondering, are these fragments then secreted, which can actually play a role in disease propagation? So for this purpose, we switched back to HeLa cells because it requires a lot of material to really isolate the medium and then also check the vesicular structures in the medium versus the soluble structures in the medium. And what we did a few centrifugation steps, followed by size exclusion tomography, which fractionates the different medium fractions into an extracellular vesicle fraction, in which we propose that the APP-CTFs might be detected, and a soluble fraction in which we possibly can detect SAPP, which is also like one of these fragments that gets secreted upon APP processing. On the left, you can see cell lysates, the extracellular vesicle fraction and the soluble fraction. And what you see in the cell lysate fraction is that you have increased APP-CTF production, again, in the Hobbs knockout cells. If we then focus on the extracellular vesicle fraction, we see that there is no full-length APP to be detected in these extracellular vesicles that are secreted into the extracellular space. However, we see also a huge accumulation of APP-CTFs in Hobbs knockout cells in this extracellular fraction. And also in the soluble fraction, we see increased secretion of this soluble fragment in Hobbs knockout cells. So to confirm this finding, we also did some imaging approaches. So in brief, what we did, we coupled APP to fluorescent protein, which is pH sensitive, and we overexpress this with an extracellular vesicle marker, which is CD63. And that one we also coupled to a pH sensitive probe. And what we see upon fusion with the plasma membrane, the there would be a pH shift. So normally the fluorescence is quenched in endosomal compartments. However, upon fusion with the plasma membrane, there's an elevation in pH, and therefore you can see fluorescent signal. And this is also what we see in the video on the right. So we see multiple events where you see both CD63 and APP flashing at the plasma membrane, indicating that these fragments are secreted via extracellular vesicles because they are positive for this extracellular vesicle marker CD63. So, as take-home messages, I want you to remember that APP normally is present mainly in the Golgi and in early endosomes, and that APP normally is able to recycle from these early endosomes back to the TGN. Also, APP can be degraded in lysosomal compartments. However, if we look into Hobbs impaired cells, we see that this recycling pathway is impaired, and that APP and base 1 then can accumulate within the same late endosomal compartments, upon which we see increased formation of neurotoxic APP-C terminal fragments. And lastly, what I want you to remember is that upon this dysfunction of endo-lysosomal fusion by Hobbs disruption, we get increased secretion of APP-CTFs via small extracellular vesicles called exosomes. So I would like to finish by thanking the Farias lab and the Klumperman lab for their assistance during this project. I would also like to thank the Verwij lab for their assistance with the EV work, the technicians in Judith's lab for the help with the EM, and of course the funding from Alzheimer Nederland. And with that, I would like to thank you all for your attention. Thank you.